Welcome to my lecture on photoreceptors. Here I'm going to talk about the structure and the electrical activity of specifically the photoreceptors. One thing that's very important as you study the visual system, you've got to remember where you are. You've got to remember whether you're in the retina, the geniculate, the striate cortex, or somewhere else. So again, right now, last time I went over a quick overview of the retina as a whole. Right now, I'm going to spend some time focusing on the photoreceptors and then the flow of information to the brain. This is a scanning electron microscope of a monkey retina. You can see that there's two different types of cells in the body. These are not colored. This is computer enhanced. But you can see the shape is very, very different. Consequently, these are called rods, and you can see these are kind of cone-shaped, so they're called cones. Right? As you may have imagined, the name rods and cones are based on their structure. That came long before we could study electrophysiology of them. Now, when we look at the rods, again, I told you there's two parts of a photoreceptor. There's going to be an inner segment and an outer segment. It's the outer segment that has the photopigment. This is what responds to light. The inner segment has a nucleus. It makes all the stuff that allows this to operate. That's going to be true whether we're talking about rods or cones. Both rods and cones have an inner and an outer segment. The main difference between cones is not and the rods is not the inner segment. It's actually the outer segment. And you can see rods have a much bigger outer segment. Gives them more photopigment, may be part of the reason why rods are more responsive to light than cones are. So just in general, this is, I'm sure, review. They're highly sensitive to light. They're easily bleached. And what I mean by that is that they can run out of photopigment. Now, what do you think happens if a photoreceptor were to run out of photopigment? Remember, no receptor. We can't detect the stimulus. So when we run out of photopigment, now we can't detect light anymore and functionally become blind. Right? This happens in rare circumstances. In the human retina, the rods are going to be concentrated in the periphery. These have no color information. So again, these are going to be black and white. One of the things that rods are really good at also, not because of them, but because of the circuitry to the brain, these are going to be really good at detecting movement. Okay, so again, sometimes you'll see something move out of the corner of your eye. That's going to be rods, not cones. And again, it's not the rods themselves that detect movement, right? Movement is a perception. But these are hooked up to a neural circuit that allows you to detect movement easily. Now, the rods have a photopigment, a chemical called rhodopsin. When rhodopsin is struck by light, we get a splitting of the molecule into opsin and retinol. Okay, so I do want you to remember rhodopsin. This is the photopigment of the rod. Light strikes it, and it splits it into two chemicals, opsin and retinol. You need to remember those. You do not need to remember the chemical structure. Now, when the light hits this, we get a very quick reaction. Okay, so here we have rhodopsin. Light hits it. It's going to induce a shape change. When that happens, we end up getting opsin and retinol. Okay? Now, this conversion is going to release energy. Okay? And I'll talk in a minute about how this energy that's released by this chemical reaction, triggered by light, but it's a chemical reaction triggered by light, that is going to be transduced into a change in the membrane potential of the photoreceptor. Okay? This occurs real fast. Now, we need enzymes, we need some energy, and we have to have vitamin A around. If we have those things, opsin and retinol, we can rebuild our rhodopsin molecule. But that happens more slowly. So think about if you're in a situation where there's really bright light. It's hitting your retina. It's hitting the rods very quickly, or very strongly. We're getting a tremendous conversion of rhodopsin into opsin and retinol. The splitting is occurring constantly and at very high levels because the light is so bright. Eventually, we're not going to be able to replace the rhodopsin as quickly as we're using it. When that happens, again, you can experience temporary blindness. Once the reactions catch up and you make more rhodopsin, now you're not blind anymore. In a tropical environment like Singapore, I don't know what situation you might see this, but in like the Arctic, people have to worry about snow blindness. So the glare from the snow, so much light strikes the retina that this can occur, and eventually you can't see. Now, in terms of the transduction, I don't want you to memorize this in detail. Okay? But when light hits the rhodopsin, it's going to split it, and it's going to activate an enzyme complex called transducin. This transducin is going to start a biochemical cascade. Again, don't memorize the specifics. Biochemical cascade is good enough. When we alter this function, we're going to change activity of enzymes just like second messengers in other places. That's going to activate another enzyme. This enzyme is then going to affect activity of sodium channels. Now, one thing about the photoreceptor that's a little screwy. At rest, just sitting around, doing nothing, these sodium channels are always open. What happens when a sodium channel is open? Sodium's going to flow down its concentration gradient from the extracellular fluid to the intracellular fluid. Now, when sodium comes into a cell, what happens to the membrane potential? It depolarizes it. Okay, so my photoreceptor, at rest, just normally sitting around, I've got sodium flowing in. It's always going to be depolarized. The membrane potential is always depolarized. Now, when light hits my rhodopsin and starts this biochemical cascade, it ends up shutting those channels. So, if I shut those channels, sodium no longer comes in. What happens to my membrane potential? So without sodium coming in and depolarizing it, right, my membrane potential is going to be more negative. Okay, so this is where things get a little screwy. Light hitting my photoreceptor results in a hyperpolarization. Now normally we think of a stimulus activating neurons, right? That's not what happens. Okay? Light's going to inhibit, it's going to hyperpolarize the photoreceptor. Now, this is why I explained the mechanism. That makes no sense until you understand what the splitting of rhodopsin does is eventually close sodium channels. Okay, when we close those sodium channels, now we end up essentially getting a hyperpolarization. This is just another way of showing that same thing. Normally, my cell is just sitting there. I've got sodium coming in. Okay, the membrane potential in the dark ends up being about minus 40 millivolts. Okay. Now, with photoreceptors and bipolar cells don't have action potentials. And remember, the flow of information goes photoreceptor, bipolar cell, ganglion cell. Photoreceptors and bipolar cells do not have action potentials. The ganglion cell is the first cell in this visual pathway that has an action potential. And it's important for you to remember. Now, the photoreceptors do, however, release a neurotransmitter. And the release of neurotransmitter is proportional to their membrane potential. So the more depolarized a photoreceptor is, the more neurotransmitter it releases. Okay? So when I'm here in the dark, at rest, I got my sodium channels open, sodium comes in, depolarizes my membrane, lots of neurotransmitter gets released. when light hits, now we get this biochemical cascade. That sodium channel closes, and my membrane potential becomes hyperpolarized. Now, when I'm hyperpolarized, what happens to my release of neurotransmitter? It decreases. Okay, so in the dark, I'm releasing a lot of neurotransmitter. Light hits me, now I get hyperpolarized, and I release less neurotransmitter. This is counterintuitive. I strongly suggest you draw it out and talk yourself through it, because at first glance, it doesn't make sense. It's very different than what you're used to. Okay? Again, just another way of showing the biochemical cascade. We know a lot about this pathway, about the molecular biology. I don't want you to remember phosphodiesterase, transducin. You don't need to remember all that. All you need to remember is when rhodopsin gets split by light energy, we get a biochemical cascade that closes these sodium channels. That results in a hyperpolarization. That hyperpolarization means less neurotransmitter being released. Okay? Now, that kind of still doesn't really make sense. How does reducing the amount of neurotransmitter I'm releasing at the photoreceptor eventually result in an action potential at a ganglion cell? And that's where I'm going to put some of this stuff together for you. So the photoreceptors are always depolarized. Okay, so they're always active. They're always releasing neurotransmitter. It turns out the neurotransmitter released by the photoreceptors is glutamate. Okay, so I want you to remember glutamate's always being released. Now normally we think of glutamate as being excitatory. It stimulates cells. Here that's not the case actually. The receptors on the bipolar cells respond differently to glutamate than they do in your brain. So when glutamate hits a bipolar cell, it actually inhibits it. It hyperpolarizes the membrane. Okay. When light hits the photoreceptors, I get a hyperpolarization. So the photoreceptors slow the release of glutamate. Okay, so if glutamate's normally inhibitory, and I slow down that inhibition, what happens at the bipolar cell? That's something we call disinhibition. I'm removing the inhibition. The best analogy I can think of, if you've got a classroom of young children, normally they're just going to sit and they're going to talk. But if the teacher goes shh, shh, and keeps reminding them, shh, you need to be quiet, you need to be quiet. Right? That's basically my photoreceptor releasing glutamate. Shh, shh. When it does that, what happens? The kids kind of are going to quiet down a little bit. Okay, so think of the photoreceptors as constantly inhibiting the kids talking. Now, what happens if the teacher stops shushing the class? If you stop doing that, eventually the class is going to start to talk more and more and more. They're no longer inhibited, they're disinhibited. So when that happens to the class, right, they're going to be excited. Okay? So essentially this is what's going to happen. So when light hits the photoreceptor, I get that release, slow in the release of glutamate. Without that inhibition, so with disinhibition, now the bipolar cell is excited. Now I told you bipolar cells don't have action potentials either. Essentially, they work like the photoreceptor in terms of the release of neurotransmitter. So if my bipolar cell is depolarized, it's going to release more neurotransmitter. If it's hyperpolarized, it's going to release less neurotransmitter. Okay, so my photoreceptors are always shushing the class, releasing glutamate. That is going to inhibit, hyperpolarize those bipolar cells, so they're going to release less neurotransmitter. When those bipolar cells release less neurotransmitter, the ganglion cells are not as excited. Okay, so what that means, the neurotransmitter release between the synapse between the photoreceptor and the bipolar cell is inhibitory. Bipolar cell and ganglion cell is excitatory. Okay? So when light hits, less glutamate, I get disinhibition, so my bipolar cell is going to release more neurotransmitter. What effect does that have at the ganglion cell? Now we're kind of back to normal, like I said. So more neurotransmitter, more excitation. So that's going to excite the retinal ganglion cells, will exceed threshold, and will fire action potentials. Okay? So here's a diagram kind of putting everything I just described together. I got my photoreceptor, bipolar cell, ganglion cell. This ganglion cell is going to send an axon onto the brain. Here, we're going to show membrane potential in the case of these, and I'm going to show action potentials in the case of ganglion cells. Now, remember that as membrane potential goes, I have a parallel change in the release of neurotransmitter. Okay, so when my stimulus, light, hits my photoreceptor, those sodium channels are going to close, and what happens? I'm going to get basically a hyperpolarization. With this hyperpolarization, what happens to my release of neurotransmitter? It goes down. Okay, so I'm getting less and less neurotransmitter being released as I have that hyperpolarization. Okay. With less inhibition, okay, with disinhibition at the bipolar cell, now the membrane potential here is going to depolarize. As my bipolar cell depolarizes, what happens? Releases more neurotransmitter. Turns out it's glutamate also. It's going to release more glutamate. That's going to excite this ganglion cell. If we exceed threshold, we're going to get action potentials. Okay, so to make a long story short, we're going to have graded potentials here. We'll have action potential here. Light is going to hyperpolarize the photoreceptor. That results in disinhibition or depolarization of the bipolar's membrane cell potential, release of neurotransmitter, action potentials to the brain. Now be able to draw this out, because on an exam I'm going to ask you what would happen if I hyperpolarized the membrane potential of a bipolar cell. It's a good question. What would happen if I hyperpolarize this? What's going to happen to my release of neurotransmitter? It's going to go down, right? If I have less neurotransmitter being released here, what happens to excitation of this ganglion cell? That's going to go down too. So what's going to happen to action potentials? I'll have fewer. Okay, so if I do that, you're going to perceive less light information. Okay? Think about what would happen if I took glutamate and somehow I could just squirt glutamate here onto the dendrites of the bipolar cell. What would a person perceive if I gave him glutamate right here? Again, walk yourself through it and see whether you come up with the right answer. All right. That's the basics of rods. Now, cones pretty much work the same way. So I'm not going to go through the details of transduction with the cones. For the purposes of this class, just know that it's very similar to what you see with the rods. Cones use different photopigment, so instead of rhodopsin, they have other photopigments. Okay, but you get the general idea, the concept. Light hits, splitting, closing of sodium channels, hyperpolarization. Okay? So I'm going to spend time talking about the cones. The cones are concentrated around the fovea, as you know, and they're relatively insensitive to light. Without cones, we don't get color vision. Now, one way you can show yourself that they're relatively insensitive to light, if you sit in a room with a dimmer switch, okay, and you see all the colors, and you turn down the lights a little bit, eventually you're going to get to a point where you turn down the lights enough that the cones can no longer respond. You can still see the room okay, but you can't see the colors anymore. Okay? That's because the cones need quite a bit of light in order to respond, whereas the rods don't. All right. Now, when we talk about cones, they used to be called red, blue, green. Now, more properly, we're going to use long, medium, and short wave. Now, remember, I did ask you to remember those numbers in terms of human visual spectrum. So short wave, what wavelength are we talking about? Hopefully you remember 410. Here we're talking about violet. Long wave, about 770, red. Okay, so basically red, green, blue is what they used to be. Okay. Sometimes I'm going to slip and still call them red, blue, green, but more properly, and I want you to remember, I'm talking about long, medium, and short wave cones. Okay, now, what I've just described is basically how the photoreceptors signal that light is present. We're going to have to take that information, transmit it along a neural pathway to the cortex, and then the cortex is going to have to make sense of it. Until the cortex has processed it, you don't know anything about it. You don't know whether it's bright light or dim light. You don't know whether the object's moving or stationary. You don't know what color it is. That's all a function of the cortex. And I'll talk about those processes individually in future lectures. Right? Thank you.